So, genes are not the blueprint for life. And that is the title, believe it or not, the top science journal in the world in February of this year published an article with precisely that title. They knew exactly what the editors were doing because, in effect, they were announcing the end of 80 years at least of misunderstanding the fundamentals of biology. And this talk is going to reveal to you why that statement is true. Because standard biology, I'm sad to say, because I'm a biologist myself, taught students over 60 years now, has got causation upside down. Genes, to quote Richard Dawkins, genes create us, body and mind. I reply, we control them and change them when we need to. So the structure of the talk is going to be that since, as I will show, genes are not the blueprint of life, measuring them and their association scores with common diseases, which is something biologists do a lot now, cannot work for the purpose for which it was intended. And I'll first explain why that is the case, and then conclude by showing that statistically, anyway, with a study from University College London, it doesn't succeed even in predicting common diseases. And since that approach fails, and that's the reason why the IAI has asked me to come and give this talk today, what is the alternative? What can biologists now do to deliver what we've failed to do? And the answer is going to be to switch to investigating the functional networks in organisms that control the genome and enable it to be edited. And to show how that can be done, that there is hope on the horizon, I will choose to close with two very interesting examples of what physiologists can achieve. So, just a little bit of 101 biology. What exactly is the genome? For those of you who need to be reminded, it is, of course, a very long, thin thread of molecules called nucleotides because they're in the nucleus. And there are four types. You don't need to know the actual names, A, T, G, and C. And in humans, you and me, and many other animals too, there are at least 3 billion nucleotides. That's an important figure for a bit later in the talk. All our cells, except for the red blood cells, contain a complete set of those. Now, the important thing to realize is as chemicals, because that's what they are, they can only do what chemicals naturally do. Their associations and dissociations from other chemicals in the body are determined by the energies of those associations or dissociations. So A likes to be with T, G likes to be with C. And in doing this, they have no choice. And that is the fundamental reason why it's incorrect to describe them as selfish, either metaphorically or literally. Only organisms with the freedom to choose can be described as selfish or cooperative. A baby, therefore, is not born selfish. It simply has needs, food and care to enable it to live, to grow and to flourish. And it slowly learns that it can choose. And after about 3 or 4 years, the parents get fed up, of course, because by God, by then it can choose and it knows what it wants. But for those few years, it doesn't. So let's get down to the nitty-gritty. Why is looking at the level of genes not the best way to get to understanding a living system? Well, I would say it's because focusing on the genome sequencing, and that alone, is like mistaking the pixels from the message. This is the message at the end of the little book, Understanding Living Systems. And if I expand it up by a few tens of times, you can see the pixels, but at sufficient magnification, it's no longer possible to see what the message is. And this is my analogy for explaining why, yes, genes are important. We would not be able to make the proteins we make that enable us to move and do all the other things that organisms do. That's not my point. Of course, genes are important in enabling all that to be done. But nothing right at the top of the organism really depends on being able to read the A, T, C, and G. Does your medical practitioner, your GP, read your C, T's, A's, and G's in order to decide what to do with you? No, and never will. So I asked the question, you see, how then did the genome become described as the book of life, creating us body and mind, to repeat the mantra of my friend Richard. I call him my friend because I've known him for 56 years. We debate, as some of you know, here in Hay on Wye. His books are absolutely brilliant, extremely convincing until you understand where they go wrong. But now, actually, we agreed on that 2 years ago. Some of you will remember we both told each other, your books are brilliant, but they're wrong. Anyway, now, amongst other things, I am a computer programmer. I learned to program valve computers 61 years ago in order to work out the rhythm of the heart. But if, as a computer programmer, you look for where the if, this, then that, else the other, where those clauses lie in the genome, you will never find them. Nobody has ever found that kind of computer logic within the genome. There are switches in genomes. Each gene has an area near it usually, which is a switch. But those switches are controlled by other physiological processes. They're not controlled by the genome itself, except indirectly, because some of the rest, which doesn't code for genes, codes for what are called little RNAs that do, in fact, have influence on what the genome is. But that gets you into what I'm going to refer to at the end, the physiological networks that actually control what is happening. So, where are the control mechanisms? Now, this is a very complicated diagram. I'm not going to take you through it, but it shows you just how complex a cell in your body really is. All of those structures you see there are actually lipid membranes, like soap bubbles. And it's essentially the fats in us that are forming those membranes, and it's the proteins in those membranes that have the sensitivity to the outside world, the chemicals we encounter, the electrical changes we encounter, the mechanical changes we encounter, absorption of photons, you name it, they sense it. And those are where the if, then, else clauses lie. And, of course, as I will show at the end, they send messages down to the genome to tell it what to do. Incidentally, choice is an essential element in any theory of how organisms are able to make choices. And it's important to know that in addition to all of our cells, nerve cells particularly have these controllable on-off switches. Now, there are no genes coding for membranes. Yet all of that is inherited. At the debate 2 years ago, Richard told me, Dennis, we could inscribe your genome in granite, keep it for 10,000 years, and then in 10,000 years, we could recreate Dennis Noble. I said, no, you can't. Why not? Oh, Richard, where would you get my mother's egg cell as it existed in 1936? And that's the point. You see, all of that has to be inherited in addition to the genome. So, now I'll just go through the simple proofs that were featured in the Nature article in February, that the idea that gene-centric biology is the whole story, the complete story. It's an important story. Remember what I said earlier on. Genes are important. They make proteins. They enable those to be made. But with encountered, unfortunately, 4 major errors. The first one is what is called the central dogma of molecular biology, formulated by Crick in 1958. And it's the idea that you simply go from DNA to make an RNA, or RNAs in plural, and enable the organism to make proteins. That is absolutely correct. If you keep the central dogma as a simple chemical statement of the sequence described there, absolutely correct, and you cannot reverse that. That's why it's called a dogma. It is impossible to reverse it. You can't go from proteins to make DNA. But that is not the way in which organisms control their genomes. What was happening during the pandemic illustrates this perfectly well. A new virus appeared. Our immune systems detected it. They then proceeded to mutate the relevant part of the genome, and only that part that codes for the grabbing part of the protein they've got to make, called an immunoglobulin, that can grab the virus or the new bacterium, whatever it is, and neutralize it. How do they do that? A message goes to those cells that can do that to start proliferating, and through chance changes to change the genome sequence. The system then, not the individual cell, can't know that, but the system then decides which cells succeeded in getting the one that grabs the virus. Those are told to reproduce as fast as possible, and then the rest are told to die. So the central dogma does not prevent that kind of process occurring. It clearly occurs. It occurred during the pandemic to all of us. The second dogma is one that goes back a very long way indeed. It was formulated by the German geneticist in the 19th century, August Weismann, in 1883, just a year, incidentally, after Charles Darwin passed away. Fortunate for him, because Charles Darwin would never have agreed with him. His idea was what we now call the Weismann barrier, which is that not only are the germline cells, that's the future egg and sperm, kept separate from the rest of the body, but there's a barrier between the two so that the body changes can never be communicated to the germline. Incidentally, Darwin totally disagreed with Weismann about this. Now, we've now found that all cells in the body extrude tiny packets, little vesicles, if you like. They're almost like very tiny cells, but they're not really cells. They're just vesicles containing some of the control molecules, RNAs, proteins, and other molecules that control the genome. Many of those bubble over and pass across the so-called barrier and have now been shown to affect the metabolism and other major physiological properties of the germline cells. Incidentally, if you doubt any of this, all the references for that process are in the transcript of the debate with Richard Dawkins, which you can find online. The third one is one that I also discussed with Richard Dawkins 2 years ago, that DNA self-replicates like a crystal. That idea goes back to 1942 and the lectures given in the Institute of Advanced Studies in Dublin by the great quantum mechanics pioneer Erwin Schrödinger. He had a very good idea. He realized that genetic material, the information in the genetic material, would have to be stored at a molecular level because it is so vast. So he argued, and these were the days when physicists were working out the structure of very complex molecules in the body by using X-ray crystallography. You shine X-rays through a crystal and it diffracts the signal, and from the signal you pick up, you can reverse engineer to find out what the structure was that caused that diffraction. That's how physicists many years ago first were able to work out the structure of biological molecules. That process of self-replication, which is what he proposed must be the case because it obviously replicates, is accurate up to a point. the A's and the T's and the C's and the G's like to be with each other. So once you unravel the double helix, that will automatically happen. But like all chemical reactions, it has an energy of formation and dissociation. And that enables you to calculate and indeed to determine also the error rate at which that process will occur. It's 1 in 10,000 base pairs. Doesn't sound very much. If you wrote an article with 10,000 words in and only one word had a typo, you'd feel pretty pleased. But remember what I said at the beginning. We have 3 billion base pairs. There would be hundreds of thousands of errors with that process. What happens? Each time a cell divides and forms 2 daughter cells, both daughter cells get an almost completely accurate copy of the genome. How can that possibly be? The cell, the living cell itself, has an army of cut and paste enzymes which it controls to achieve the correction of virtually all of those errors. You end up with an error rate of only about 1 in 10 billion, which is extraordinary. Schrödinger can't be blamed for his idea. He had no idea this was possible. I think if anybody had suggested to him in 1942 that this is the way it works, he would have hardly believed it. And therefore, since self-replication of DNA is impossible, you need the cell to have that organized army of cut and paste enzymes to replicate. The replicator cannot be represented as separate from its vehicle. That's the fundamental thesis of the selfish gene, and is the reason why 2 years ago I said it's incorrect. So the correct interpretation of the molecular biological evidence shows that all of these dogmas are incorrect. That's in the Nature article published in February of this year. Now, the implications of all of that for health and disease are extremely important. Because surely, some of you will be saying, in your minds at least, doesn't genome sequencing deliver the goods that matter? That is, new medical treatments. Find the gene variant causing the disease, then replace or delete it. Now that's fine, and indeed for many rare genetic diseases, that is the process through which many people are looking for genetic cures. That's fine, but with the exception of those, the answer is an embarrassing no. I'll come on to that in just a moment when I've explained what was promised. The head, Francis Collins of the National Institute of Health in the United States, leading the Human Genome Project in the 1990s up to the period 2001 when the first sequence was announced and published simultaneously in Science and Nature, he wrote that within 10 years of human genome sequencing it would lead to, and I quote exactly, previously unimaginable insights, and from there to the common good, including a new understanding of genetic contributions to human disease and the development of rational strategies for minimizing and preventing the disease phenotypes altogether. Well, University College London, a big team, put that idea to the test in October, or published in October last year in the British Medical Journal, to show that when you sum together all the relatively small scores of association between many genes, because most of these diseases are complex, multifactorial diseases. Remember, I'm accepting the monogenetic diseases. What that does is to form what is called a polygenic score. That is, you just add the whole lot together. The fact is that even with all of that, what that article showed, using exactly the same techniques as you would use for a clinical trial of a new drug, that it simply doesn't work for cardiovascular disease and cancer. Those two account for 60% of fatalities, and we can talk about the other ones a bit later. So that's why it hasn't happened, because what we found is that the association scores between the presence and absence of most genes and the incidence of major disease are generally very low. Now, I showed that in my own work with a team of scientists in Oxford looking at the pacemaker mechanism in the heart. There are several, and you can knock a major gene out that we know is causally important. Remember, I said that genes are causes in that sense. You can knock it out and the frequency change is very modest. So it's clear that organisms back up the great majority of the genes. And now I come to the end of the talk. So what do we do now? That's the big challenge that I think biologists now face. Well, what we need to do is, instead of focusing on this level where it's extremely difficult to see the message, we need to go to where the message is actually created, which is in the physiological networks that control what is happening. In other words, we have to function on that. We have to focus on that. And I will finish with, first of all, a statement of how difficult this work would be, because if we enlarged a single nucleotide to the size of my fist or the size of a golf ball, the nucleus of the cell would be way down here in Hay, and the edge of the cell would be way up there in Scotland. And believe it or not, the cell knows how to transmit information from the outside world to the nucleus, and it does that through extraordinary fine, what are called tubulins, little roadways, if you like, that enable messages to be transmitted down to the nucleus. And I'm going just to highlight two studies that show that. This is from the work of my own colleague Dick Chen, who worked with me as a research student many years ago. They, at New York University, showed that calcium, that's one of the ions involved, entering the surface membrane creates a messenger molecule that attaches itself to one of those motors, goes all the way down to the nucleus, and then controls the gene expression level. The second one is from the work of Professor Parekh in my own department in Oxford, where he and his colleagues showed it's possible for two surface membrane sites to be activated to send two signaling molecules, again traveling on the tubulins and controlling gene expression. So I finish with my challenge to the world's scientists. Those two groundbreaking discoveries show that the functional tubulin pathways from the surface down to the nucleus exist, and that they can mediate what we might call electrotranscription coupling. That's changing gene expression. So it's a long expression that tells you that's what is happening. Question, who can achieve the same for what we know happens in the immune system and in other systems in our bodies? How can electrogenome editing coupling work so that one can see what changes the genome when we need to do so? I have no role in deciding who gets Nobel Prizes, even though my name might suggest I do, but I think a Nobel Prize is awaiting for anybody who can achieve that particular challenge. Thank you very much. That's the end of the talk.